Is it okay for everybody? Okay. Thank you, Neil, for the introduction and also invitation, and also to the organizing committee, above all, Beat. I was here around 5 years ago and also presented on the technology binding albumin prodrugs to circulating albumin. Of course, at that time, we didn't have so much clinical data. This is going to be a talk about the clinical update of Aldoxorubicin, but I'm going to start with some data on preclinical studies connecting basically to Neil's talk. So I'm going to take you back in time to 1986. A young Japanese scientist does a very simple experiment. He injects this dye called Evans blue into a tumor-bearing mouse with a subcutaneously growing sarcoma. And to his surprise, already after 6 hours, he observes the blue color accumulating in the tumors, which intensifies with time. Now, what was the reason for that uptake? Was there an affinity for the drug or for the dye? In this case, for the tumor. The reason was somewhat different. This dye has a high affinity for circulating albumin, so once it's injected, it binds physically to albumin, and albumin transports the dye into the tumor. And this is Hiroshi Maeda, who did this experiment. He then looked at this in more detail, and when you look at the profile, this is the Evans blue albumin complex from this experiment. A high circulation time going down. In the tumor, you can see the uptake peaking around 48 hours, and as a comparison, in the muscle, you really don't get any uptake. The explanation for this was, as most of you know, was the EPR effect, the enhanced permeation and retention effect. I think you are all acquainted with that, but he coined this expression and is the founder of the EPR effect. It's actually most fascinating when you do this experiment yourself. This was done about 6 years ago in my team, and this is identical 10 milligrams per kilogram to a nude mouse. You can then take off the plasma, dilute it, centrifuge it, and these are the blue colors you see, and these values are identical to those found by Hiroshi Maeda. It's a very reproducible experiment. The EPR effect was discovered in 1986, published in Cancer Research, but basically neglected for at least 10 years. It was then picked up by the field of polytherapeutics and later by what we now call nanomedicine. So basically, this protein albumin is the basis for drug deliveries relying on macromolecules. If you do the experiment in a tumor-bearing animal, the tumors turn blue. This is after 2 hours in a pancreatic xenograft model. And if you open the mouse after 6 hours, you see all of the organs are blue. You have to infuse, you have to perfuse for 3 minutes with saline to actually get a picture of where the Evans blue albumin complex is going. And it's going there. This is the pancreatic tumor of 3 of these mice, and you can see they are stained blue. This is the liver and kidney, and when when you extract the amount of Evans blue from these pancreatic tumors, this is what you basically see. There's an increasing amount of blue color. 24, 48 hours would be the maximum. So you basically get uptake of Evans blue albumin complex in these tumors, and they actually still remain after this color at 186 hours. Now, this is physical binding to albumin. So in a way, it's comparable to the Abraxane technology, but of course different. But if you take the picture that Evans blue albumin is binding to various sites on albumin, this is the carrier for that dye as a model compound for paclitaxel. These are now the differences to Abraxane. With this technology, it's a Prodrug technology where you have covalent in situ binding of prodrugs to circulating albumin. So we're not using exogenous albumin, we're using the circulating albumin of the individual organism. We have prodrugs and we have covalent binding. This is what we develop in the lab. Prodrugs, which consist of a protein binding group. They then have a predetermined breaking point, a linker, and this is cleaved either by an acid-promoted mechanism or enzymatically, and we have the drug. This is injected intravenously and binds basically instantaneously to the cysteine 34 position of albumin. So this is a low molecular weight prodrug, it becomes a macromolecular prodrug. This then circulates with the benefits that the drug doesn't diffuse into healthy tissue, and with time you get uptake in the tumor, where you then have the release. And this is the drug we then developed and fine-tuned for the first clinical trial. Chemical name is 6-malamidocaprylhydrazone derivative of Doxorubicin. In its beginning years, it was abbreviated Doxy-EMCH. Now it's Aldoxorubicin. So you see the melamide group here. This is the linker. This is the predetermined breaking point at the C13 carbonyl group of Doxorubicin, and this is the well-known drug Doxorubicin. On the right-hand side, I'm showing you how it is now used in all clinical trials. It's a commercially viable drug, reconstituted in the clinical pharmacy, diluted in Ringer's solution, and then there's a half an hour infusion to get it into the patient. Preclinical data had already shown, meanwhile now in 10 mouse tumor models, superiority over Doxorubicin. In 50% of these models, we had observed complete remission. This is the decisive amino acid on this circulating protein, which of course is the most abundant protein in our bloodstream by far. And the thiol concentration at this position accounts for 90% of the thiol content in blood. We have around 500 micromolar of thiol in concentration in the blood, and that accounts for 90%. That's why you get this rapid binding to the circulating albumin. The mode of action after tumor uptake by the mechanisms very similar to probably Abraxane, EPR effect, GP60 receptor, spark. Once it's in the tumor environment, two modes of release. It's known that the extracellular environment of tumors is acidic between 6.6.8. You can measure that with pH electrodes, so there is a chance of extracellular release with then diffusion of doxorubicin into the tumor cell. Most of it will be taken up in endosomes or lysosomes where you have a pH of 4 and 6, and then doxorubicin is cleaved. It comes out of the endosomes and lysosomes and then has its different modes of action, DNA intercalation, inhibition of topoisomerase 2, resulting in apoptosis. The aldoxorubicin positioning. Doxorubicin is the best-in-class anthracycline. One very important factor, we've treated over 400 patients. Meanwhile, we've seen no indication of acute cardiotoxicity, which of course is a severe toxicity for doxorubicin. We have the advantage that we can dose higher. Doxorubicin is dosed as 40 to 75 milligrams per square meter. This is the curative dose, and in dox equivalents, we dose at 170 to 260 milligram per square meter. This is up to a 3.5-fold increase in the doxorubicin amount we can give, but even more, we can give more than the 6 traditional cycles. So some patients have, for instance, received way over 20 cycles. There is an improved quality of life. Alopecia is less severe and not as frequent as doxorubicin. If we go down to the lower dose here, 170, we basically only see grade 1 or grade 2 toxicities. This is an overview of the clinical development program and the trials which are ongoing. We have one registrational trial, a phase 3 against second-line soft tissue sarcoma. We have an almost finished phase 2 study in first-line tissue sarcoma. We have orientating studies in glioblastoma, multifoma, and Kaposi sarcoma. We have a larger study, a comparative study to topotecan in small cell lung cancer. This is second line. Pharmacokinetic study completed, and we have 2 combo studies going on with ifosfamid and gemcitabine. This is in solid tumor, this is in sarcoma. Why soft tissue sarcoma? This was the one we concentrated on first. Doxorubicin and ifosfamide are the standard agents for treating this disease. So we have the chance of comparing this drug to aldoxorubicin. And you'll see later that this gave us a chance to establish a superiority over doxorubicin. Why second line soft tissue sarcoma for registrational trial? This is a very hard to treat disease. There's no standard care for most patients. When you look at the endpoints you have to reach, progression-free survival is an acceptable endpoint, and that shortens the time for completing such a large phase 3 study. You do not need to show superiority for overall survival, and we had preliminary data showing that in these kind of patients we had good response. I'll show you that later. The setup of this study was we had all in all 123 patients. They were randomized. We had 83 here and 40 here. They were giving the maximum curative dose for aldoxorubicin every 3 weeks, 260 milligram per square dox equivalents at 350 milligrams per square meter if you take the mole mass of Aldox into account, compared to the curative dose for Doxorubicin, both given every 3 weeks. Oops. Oops. So this is the data for the progression-free survival. I've highlighted the difference, statistically significant. We had an improvement over dox of 3.7 months between the two arms. These were the scans read by the investigator. This phase 2 trial, was designed to have an assessment by the investigators in over 80 centers, and it was also read independently by a central lab, and there the benefit was basically 3 months, also statistically significant. So we moved on to a phase 2 trial design. No? Am I right? Got it? This thing is not responding properly. Why do I have to hit the button to get it on? Okay. Overall survival. Overall survival in this study was, it's not totally completed, in the this is the result from the investigator-initiated analysis, 16 versus 14.4 months, so that's 1.6 months benefit. If you look at the patients surviving over 800 days, it was 40% for Aldox, and for Doxorubicin 20%. And this is the data, if you look at the patients who hadn't received any chemotherapy, it was 2 months, so somewhat higher. It just doesn't move on. Go on. Strange. Okay. Seems to be going for, I'm pressing the same button. This is now still the phase 2 study, and this is giving you a picture of response rate over these patients of the 83 to the 40. Overall shrinkage, and that's of course these patients, 60.8% compared to 39.4% for Doxorubicin. If you take the objective overall response rates, which have to have a remission over 30% by tumor CT scans, that's this part. Some are nearly reaching complete remissions. And as you can see here, in the Doxorubicin arm, there are no objective responders. And these, of course, are the patients which have benefited the most from this study. Try it again. No? Well, I have to point that way. Good. Phase 3. We moved on to the phase 3 study. It was a randomized comparative trial design. It had a special protocol assessment granted by the FDA, which allowed for dosing until disease progression. The reason that this is a very hard, difficult to treat disease. The patient population, they all had progressed following treatment with chemotherapy, 400 patients in 2 arms. And they were allowed to have received Doxorubicin or a related anthracycline. The endpoints, progression-free survival, the secondary endpoints, overall survival, response dates, and safety. Go. Yeah, this is strange. Okay, this is some data we had before, why patients who had already received Doxorubicin had not responded, or another anthracycline, responded in a small trial when they received 260 square millimeters of Aldoxorubicin, and these are these patients. There were 5 of the 13 patients who responded very well, so that was encouraging data with these long progression-free survivals and overall survivals for these patients. Let's see what happens now. Strange. Can you hit the button? Where should I point this? Over there? Okay. So I have to really point it over there. Okay, so it's not getting the signal. No, it's going back again. No, that's forward, right? That's forward, right? This is back. It's that one. Turn it? How can I turn it? Press another one. Another one. That's fine. Okay. Which one are you pressing? Yeah, that's the one I'm pressing.
Should I just do it for you?
Oh yeah.
Yeah, just say
Right.
You gotta just a couple of
Yeah. Okay. Well, this has held me up. Aldoxorubicin for small cell lung cancer. This is still the most important trial compared to the sarcoma studies. Small cell lung cancer is associated mostly with heavy smoking. 15% of all lung cancers usually advance at diagnosis. The approved second line is topotecan, which is not considered as a very promising drug. They have significant toxicities from this drug, and patients rarely finish a course of therapy. If they do receive topotecan after refractory platinum situation, PFS is 3 months, 9 weeks, overall survival is 21 weeks, response rate 5 to 20%. So definitely there's a better need for treatment options. We had these were individual patients, we had some very encouraging results at low doses of Aldox. 180 milligrams per square meter produced a nearly complete response, 80 weeks survival compared to those 20 weeks. Another patient, 21 cycles, 63 weeks, and another one, 50 weeks. The trial, 200 patients in each arm getting Aldoxorubicin at the low dose, 170 milligrams per square meter, and topotecan at these regimens. The endpoints are progression-free survival, overall survival, and overall response. At the moment in 40 clinical sites. Trial started Q1 2015, and we expect to get progression-free survival data in Q2 of 2016. The reason why we're dosing at these lower doses, we had already seen very good and encouraging responses, and as you can see, there has basically, as I'd already mentioned, no grade 3, grade 4 toxicity. Short summary of the glioblastoma, 30 patients enrolled. that's the aim at the moment. These have been enrolled. We've seen tumor shrinkage and prolonged stable disease in several of the first 18 patients. 3 patients had no microscopic evidence of tumor when the tumor was examined after surgery, representing a complete response. And that suggests that it crosses the blood-brain barrier. We already had seen that in 2 very encouraging animal models which have been presented. We also have 1 study going on in 30 patients in Kaposi sarcoma. These are all which have HIV. Doxil is the standard of care. We're treating at 50, 100 or 150 milligrams per square meter every 3 weeks. Results so far in 9 patients had a decrease in skin lesions and number of cancer cells expressing the virus. 6 patients with lung tumors had a complete or partial response, and no patient had disease progression. The ongoing trials, this is more or less the last slide, is just a summary. This is really looking forward that we expect to combine Aldox with other drugs. This is in sarcoma. In one site, we have 1 patient who had a very good response, a near complete response, and a number of patients who had tumor shrinkage. Aldoxorubicin and gemcitabine is being examined against solid tumors. So the concept here is we have demonstrated superiority in a phase 2 soft tissue sarcoma study. The registrational phase 2 is on track. We have at the moment actually 226 patients which have been enrolled. We're expanding with a further phase 2 trial. This is second small lung cancer, a comparative study which could get first track approval if the data is convincing. And we have ongoing combination trials, and that should set the platform for, as we hope, we're optimists to get market approval and then start to do combination trials in other indications such as pancreatic, ovarian, neoadjuvant. Just one point I want to highlight. We are concentrating on orphan drug indications for 2 reasons. These are hard to treat. And the other reason is that we have an extended patent time life for these indications. This is the last slide. It's just to show you that we started further preclinical work in Freiburg, set up a CytRx drug discovery branch, and have meanwhile extended this technology. It now has the abbreviation and it's trademarked LADDER TM technology, which stands for Linker Activated Drug Release. It's a similar system, but it allows a very controlled release of the drug, which will be important not only for the drug in question, but also for the indication which you're aiming at. And of course, these kind of prodrugs can be bound not only to albumin, monoclonal antibodies, and being at CLINAM, also to nanocarriers. Thank you very much. Sorry for all the slides moving up and down.
Thanks very much for the interesting talk. Do you have or do you plan to have PK data to support the hypothesis that you are getting more in the tumour compared to other tissues or doxorubicin? And is there any attempt to maybe even stratify tumours to look for tumours that have more leakiness or high EPR, and how would you do that?
Regarding the first question, we do, from similar to Neil in animal models. Our data suggests we have a 6-fold increase versus Doxorubicin at equitoxic dose. That's basically how we can explain complete remissions in mice models. The good thing was that we could transfer the increase in dose we had seen in mice models, which is actually 4.5, to the human setting, which was a problem with Doxil, which had to be lowered to a new side effect coming up, the skin toxicity. The second question, at the moment we don't have any plans for setting up, which is actually a challenge. How is albumin taken up and to what extent in humans?
Just, do you need to have an albumin level, a minimal albumin level of the patients for eligibility, or do you see any correlation with the albumin level and the pharmacokinetics?
No, we checked this in phase 1, how much of the albumin we would be saturating at the highest dose, and we're saturating, if we were getting 100% binding to cysteine 34, 30% of the albumin is saturated in that cycle. You have to consider this is 50% of the albumin in our body. The other 50% is in the tissue and the lymphatic system, so you're actually only saturating 15%. And then you have to consider that in every week we produce 10 grams of fresh albumin with fresh cysteine. So after 3 weeks in a cycle, you already have 30 grams. And as you know, in our body, per liter, you have approximately 40 grams times 5, so 200 grams. So this is a very dynamic system which is constantly producing cysteine 34.
So my question is actually related to the previous question. How long it takes to conjugate the maleimide group to the EMCH, so maleimide group to the cysteine? Because you could also do something like taking the blood, or taking, let's say you withdraw the blood from patient and you incubate with your Dox EMCH and then re-inject the
No,
blood.
that wouldn't make sense, because it's definitely an advantage to have the patient's own albumin. And as I said, the Michael addition of a maleimide is so fast that it's really hard actually to detect if you were injecting, let's say, Aldox rapidly, like within a minute in a bolus injection. It'll be very difficult once you take that blood off to actually show that you still have any relevant amounts of Aldox, because even while you're injecting it, there will be a slight, of course, diffusion into the tissue, but it's so minimal that, of course, you don't have 100% of it going to the cysteine 34, but I think we can confidently say that it'll be approximately 90%.
And are there more proteins with the free cysteine? Are there more proteins with free cysteine?
There are one or two, yes. There is apolipoprotein B, but that sits in a very deep cavity, not accessible. There's antitrypsin, and that's about it. And there are some free cysteines, and people think that cysteine, homocysteine, glutathione are free thiols, but they are dimers. So cysteine is present as cysteine to 95%. So those levels play a marginal role.
You didn't show any PK data. What is the clearance and the half-life of albumin, unbound free albumin, like we have it in our blood? And what is the half-life and clearance of your albumin coupled with your drug? And if there is a difference, what explains the difference?
There is a difference. If you take the half-life values from the literature for human serum albumin, it's in humans around 19 days. We have, once it's bound, the half-life goes to around 24 to 30 hours. So it changes, even just binding one albumin molecule very tightly into that cavity, it changes it. The most likely explanation is that the FC receptor doesn't recycle it, maybe the way it does for human albumin, which hasn't been modified. There's definitely no evidence that it's being drained off to the liver. And the other question was?
Yeah, that was about it. That's
Okay.
We have probably time for one or two more questions, and we can address if you have any questions on the previous talk. We can do that. Okay, I have a question for you, Felix.
Go ahead.
So you go in your registrational trial, you're going for a second line indication with you're going for a second line indication in registration with very limited second line data. Is that correct? From what I understood, your phase 2 trial that you did was a first line trial.
Yeah.
And so what do you think the risk is there? You know, you have limited data with second line patients, and then you've launched into the second line registrational
Well,
trial.
first of all, I mean, there's these patients, they really have no alternative at the moment. We know that the drug is active against sarcoma, and sarcoma is a very heterogeneous disease. So these patients will have got anything mostly from doxorubicin to ifosfamide, maybe Votrient, and then you compare that to a standard. They have a choice of 50 or 5 anti-cancer agents. I think for this indication, as I mentioned, the endpoints we are trying to reach, because it's such a terribly hard disease to treat. The benefit for the patients, quality of life, progression-free survival, a lack of side effects, if that compared to the other anti-cancer drugs in the other arm, there's a good likelihood that we will score better, and then these patients will certainly take Aldoxorubicin compared to anything else which is on the market.
And so the last question, I think I understood that you're allowing patients into this phase 3 trial who have had prior doxorubicin exposure. And can you just explain that, why you would do that?
One slide I showed, we had 13 patients where we ourselves were surprised that they were responding very well to where they had basically not responded to any anthracycline anymore, and they were getting partial remissions. Of course, that's a small population. But of course, one of the rationales and one of the reasons for for using Aldoxorubicin for patients who generally have had received Doxorubicin in the first-line setting is that Aldox can still give them some hope.
Okay, very good. Any last questions? Burning questions? Okay. Well, thank you very much, Felix, and thank you all for attending this session.